Yeah, actually, you turned right to the page. Cranial nerves. I want to talk about cranial nerves. So I want to say a few more things about cranial nerves. First, in terms of identification, give you my method of identification. So I hope people have already started memorizing the names. Okay? Whether you use a mnemonic or whatever. So once you have the names memorized in order, the important thing is just to kind of notice that they're not like randomly, you know, it doesn't go 1, 2, 3, 4. It doesn't jump all over the place. So it goes 1, 2, 3, 4, 5. We come back to the center, 6, 7, 8, 9, 10, 11, and 12. So there's kind of a pattern you want to get so that you kind of see that order in which they go in. And then my next piece of advice, which is probably getting repetitive as hell at this point in the semester, is to make smaller groups. I don't know these, I don't identify them as where one particular one sits amongst the 12. I learned them in smaller groups, and these are arbitrary groups I made up. So you can make up small other groups if you'd like. But the first 3 are right in a row. That's so I have a group of 3, a group of 2, 2 more groups of 3, and then one at the end. And so the olfactory, optic, and oculomotor are kind of in a straight line. They're really big, and you want to cross your fingers that one of them gets on the test because they're literally the easiest 3 of all of them. So 1, 2, 3 here. So that's olfactory, optic, oculomotor. My next group is a group that I've used very non-technical ways of saying where they're located because you're not responsible for specifying the origin of these things in a accurate Latin word type of way. So these next 2 I just say are on the side of the pons because from the point of view that you see it, from this inferior view, it looks like it's coming from around the side of the pons. This is the pons. Hopefully you guys get this. One of the easiest parts of the brain. Thing sticks out like a belly. And there's 2 of them, the trochlear and the trigeminal. The trochlear is tiny as hell, and I'll pin these out again. And then the trigeminal is huge. So if it's on the side of the pons and it's tiny, it's the trochlear. If it's big, it's the trigeminal. That's my really rough way of doing it. The next group of 3 that I do originate roughly at the bottom of the pons, at the bottom edge of the pons, and they're in a row going from medial to lateral. Abducens, facial, and vestibular cochlear. And so once you see that it's one of those 3s, you just, is it the most medial one, is it the most lateral one, or is it the one that's in between? So that's the way I do that grouping. The next group of 3, which is the glossopharyngeal, the vagus, and the accessory, the first 2 of those are probably the hardest of all 12. The glossopharyngeal and the vagus, which they're showing here and here as these absolutely gigantic, monstrous things. I take it back. They're going.
I have them pointed on the right.
On the right side.
What's that? Oh, okay. Here we go. Over here. So we have accessory, vagus. Yeah, here we go. Yeah, right here. These 2 guys here. They don't look anything like that. Okay. What he looks like is a whole little array of really tiny thread-like rootlets. Okay? And the very first one of these thread-like rootlets right in here is a little bit thicker, and that's the glossopharyngeal. And the rest of the little rootlets, which will be a whole ton of rootlets together, is the vagus. So the glossopharyngeal is a much smaller, little tiny nerve about the size of one of the rootlets that come to form the vagus. And I'll pin those out, and then if you guys have questions, I'll come back there and show them to you. So those 2 are really tough because literally they're right on top of each other. The next of the 3 of that group is the accessory. Again, it's right up there with the first 3. It's one of the easiest ones to do. Its origin is on the spinal cord. If they were redefining it today, it would not be considered a cranial nerve. It's actually a spinal nerve. But it got done beforehand, before they made the definitions. And so if you see a cranial nerve and its origination is on the spinal cord, it's the accessory. The last one I do by itself, which is the hypoglossal nerve, and again, this is a obscene exaggeration of what this thing is. It is tiny, wispy little thing, and you can barely see it even on the best pro sections I have. But the character there is that it sits, back here is the olive, so this is the medulla, right? So there's a little structure called the olive, looks like an olive, and then this is the pyramid, and there's a little sulcus, a little groove between those two. So if they pin out a nerve whose origin is in that sulcus between the olive and the pyramid, that's hypoglossal. So for what it's worth, that's the way I do it. I think it's much easier to have these smaller groups, and that way you come into the test and they've pinned this one right here, and you go, oh, it's one of those 3 at the bottom of the pons, and your mind goes, instead of to which of the 12, it goes to, well, that group of 3 is abducens, facial, vestibular, cochlear. Oh, it's the most medial one. Oh, that's the abducens. So, well, there was one other thing I was going to say about this, and now it's spaced it, so maybe it'll come back to me in a minute. I can't think of it now. So that's some advice on doing the cranial nerves. I also wanted to, unfortunately, I had it off. I want to talk about functions. So the functions are 67 and 68. And again, I'm just going to recommend some grouping here to help you memorize these. And the groups that I use 3 groups. There's a group of nerves whose function is sensory only. And there's 3 of them, and there's 4 that are somatic motor only. That's a group. And then the other 5, the last 5, they're just a bitch. They're kind of like a group of bitch because they have both sensory and motor, and they might even have both somatic, motor, and autonomic. So to me, it's to the quick way to do it is to learn the sensory only and somatic motor only really quickly, get them down. They're not that hard. And so now instead of being really struggling with 12 nerves and their functions, you're struggling with 5, because 5 of them are the ones that are tough to memorize. So, and the other good thing is that a lot of these nerves tell you what they do in their names, and the 3 that are sensory only happen to be special senses, and the name of the nerve tells you what it does. So the olfactory nerve does olfaction, the smell. The optic nerve does vision. So we're down to 10 on 2 of the easiest ones there. And the one that also tells you its name, is sensory only, is vestibulocochlear. Okay, and the vestibulocochlear, you'd have to know we don't do special senses until after the nervous system, but the cochlea is your hearing organ, and the vestibular apparatus is your equilibrium organ. So that one right there tells you in its name what it does. So now we're down to 9, because those ones are a piece of cake. And then the other 4 that are somatic motor only, and I always say, because I don't have to get tested on them anymore, I have to look them up. You have trochlear, and trochlear is eye muscle control. And there's one other one that is also, and there's abducens. So there's 2 of the that have exactly the same function, eye muscle control. 2 of those 4. I also would like to point out, the abducens actually tells you what it does too. What do you think the abducens does to the eye muscles, or does to the eye? So it's telling you what it does. And then, let's see if we can look at the other ones. So we have the accessory nerve, and this is one where I get to have a little bit of fun. Everybody, all you have to do is look at the function for this and think 30, and that will be the one that you will remember. And then the hypoglossal. And hypoglossal tells you what it does as well. Hypo means beneath, and glosso means tongue. So it controls the tongue muscles. Anyway, those are the groups. Then you got the rest of them. You can see the vagus has a whole list of stuff. What a nightmare to remember all that. The trigeminal. Let me explain this. You do not need to know the branch names of it, right, or the origins or exit foramina. But function-wise, you need to know all of the, assuming it's just one big nerve. So you'll look there, and they have them separated out. But if you look really closely, the sensory, if you remember that first one for the first branch, touch, pain, temperature, and muscle sense. If you remember that, you've got it. Because what's the next one? Touch, pain, right? Touch, pain, you know, the other one. So you really memorize once, and then you have to remember muscles of mastication for its motor function. Okay. So that's some advice on doing the cranial nerves. You want to have? Did I get this one? Next up, so I want to talk, Dr. Wilson told me yesterday that she would finish the brain to the end of the lecture, did she?
Yeah.
Okay. So, here's the theme. I told you, I think yesterday or the day before, that I didn't want you to waste too much time on plastic models until we did the brain sections. And what I put out here, fortunately, are pro-sections for frontal and horizontal sections of the brain encased in plastics. So you don't have to go back in the pro-sections. Just a reminder, these came from our cadaver, they belong to UCSF, they're human remains, you do not photograph them. Not photograph. And I have a bunch of horizontal slices, a bunch of frontal slices, some of them are good, some are all right. A lot of them, one side is really good and the other one isn't. So I have to go through. We also have 3 commercial ones. These are ones that were that bought, and they're all right. Wean yourself off them really quick. They're not going to be on the test. They're annotated. So we also have one that's kind of nice is that it's a mid-sag section and it's annotated. So check them out, but these aren't going to be on the test. They have the answers written. So all right. So what I want to do is give you a method. Yes.
For the last ones, are those real? The annotated ones? Are they real brain?
Yes, all of this is real.
Okay.
Yeah, it's all real. Yeah, you'll notice the difference. These ones are completely encased in acrylic here, something that we would do. The ones that we have were done by a student. They basically took some plastic and put some Kerosene with them. So I'm going to give you a method. I hope most people, everybody would write the method down. Because the one thing about, when I told you the blood vessel would talk, I said you can't just like remember these pictures and expect to go in the cadaver room and find these things and have them look like pictures. Same thing is true with this. When you can't just say, well, this piece of gray matter is always down here, because a slice right here is going to look different than a slice a half an inch back. And a half an inch back, the slice looks different. So you can't memorize these things. So if you don't use my method, you have to do a method. You have to find landmarks and know how to find the nuclei relative to the landmarks, whether you use my method or not. Because you can't, it's not something you can memorize. So let me just get started. So what I use, should I turn more lights off? Can you see the guys thing okay? You're good? Okay. So there's lots of resources on the internet, but what I use is this website called The Plastinated Brain. And I don't know if any of you guys ever heard of Body Worlds. There's this guy from Austria who came up with this way to plasticize cadavers. If you've taken this course, it's actually really cool to go to. I let you one. Travels around. Comes to San Jose a lot. Anyway, he's got a website, and what it is photographs, really nice photographs of slices of the brain. And I don't try and write down the big long thing there. Go to an internet search engine and just type in plastinated brain, and this will be your first hit. And what you'll see here is that it has frontal, transverse, and sagittal sections for you to look at, and they're all annotated. I'm going to start out with the sagittal. We only do mid-sagittal. The only one we do. We have no pro-sections that are parasagittal, so we'll just look at mid-sagittal first, and that's the section you should start with. And then we'll go back and look at the tougher thing to do are the frontal and horizontal sections. And I think it's going to turn off more lights here. Okay. So let's start out with the sagittal section. And my method involves always finding a handful of landmarks first, and then looking for things relative to that. It's not so big a deal on the mid-sag, but once we look at the frontal and horizontal sections, finding these landmarks is key to doing it. And let me just introduce the idea, which is my landmarks, the primary landmarks you can use in the brain that really work really well are the ventricles. The ventricles are these spaces that are fluid-filled in your body, and they will tell you where you are, and it will narrow down what you could possibly see. So in this case here, and honestly, this is not that great. I think that our prosections are a little bit better. Behind this little membrane here is the lateral ventricle. In and around here, the bird's heads, or sometimes they call them moose heads here. This is the third ventricle. There's a little pathway right down here called the Cerebral Aqueduct, and then down here is your fourth ventricle. And the ventricles are also really good in letting you know what embryological vesicle of the brain you're in. So let me digress a little bit. I should have done this before I started the slideshow. Do you guys know what I mean when I say embryological vesicle? I mean, and we're talking about the adult versions of it, that we're doing adult things. So you have telencephalon, diencephalon, you have the midbrain, the metencephalon.
Mesencephalon.
So you guys know that. Okay? So first off, before I get into that, make sure that when you study this, okay, and you're studying it and on a test, if they're asking about a structure, name the structure and the pin in it. The answer to that question is never myencephalon. It is never midbrain. Those are not structures. Those are regions of the brain that contain structures. Okay? So when I took the class, I learned the structures, and then you go to your textbook or your lab manual right there. You need to know which structures are in which embryological vesicle. Okay? You need to know that the cerebral peduncles are in the midbrain, and that the thalamus is in the diencephalon. But to me, that was something I could study at home. Here, it was about learning the exact structures. But I've seen this mistake made a zillion times on tests. The number of times they asked for a specific structure, and the answer from the student is a embryological vesicle. Anyway, now going back to my ventricles, is the ventricles are, you can quickly tell by which ones you can see which of those embryological vesicles are. So that thing you should get down first. Telencephalon, lateral ventricle. Diencephalon, third ventricle. Cerebral aqueduct, midbrain, and the fourth ventricle is in the myencephalon and the metencephalon. So if you can do that, and once you see that and you know what structures are in there, you really narrow down the scope of stuff. Okay, so I can see those, and then in the mid-sag section, we'll start with the easiest thing. You have the corpus callosum right here. So that's the commissural tracts that are connecting the two hemispheres of the cerebrum. There's a little membrane that separates the two lateral ventricles here, and it's called the Septum Pellucidum. Hanging at the bottom of the septum pellucidum is this kind of white thing that's kind of hanging here, a little bit thicker. That's a structure called the fornix. You guys covered the fornix? Did you do the limbic system yet? Mm-hmm. Yeah. Okay. So here's one. That's it. The fornix hangs at the bottom of septum pellucidum. If you write that down, you will find the fornix on every single prosection by using that method. You don't need any other, it doesn't matter how you cut it. And then around the third ventricle here, the primary structure that straddles the third ventricle is the thalamus. So this big old guy up here, this is the thalamus, straddles the third ventricle. And then there's kind of a beak portion of it down here called the hypothalamus, the beak of the bird. So when I think third ventricle, those are the two big structures. There's a few others. Those are the two big structures I think. I think hypothalamus and I think thalamus. That's when I think about third ventricle. Although this is not very good in this regard. Hanging off the edge here is something called the pineal gland, which is part of the epithalamus. So here's another one. Epithalamus is a grouping of structures. We only do one of those structures. Also, on the hypothalamus, there's a little bump here, and there's one on each side, and that's called the mammillary body. So part of the hypothalamus. Once I move into this region here, I'm in the midbrain. It's not, you can't see very well, but the cerebral aqueduct comes down here. So in here I have midbrain structures, and I have these two, on each side I got these two, looks like cables, and they're white matter, and they're connecting the brainstem to the cerebrum. They're called the cerebral peduncles, and that's a cerebral peduncle there. On the back side of it, I see these two bumps here, and these two bumps are part of a bigger structure called the corpora quadrigemina. And the top bump is called the superior caniculus, and the bottom one called the inferior caniculus. Those are all structures that you can see on a mid-sag section. Once you get a little bit further down, I'm down here in the pons, and I can see the fourth ventricle here. And we really don't, other than in a mid-sag, we don't do much about the pons. You can point to the pons. But it does also give me a good mid-sag view of the cerebellum. And there's two things that you need about the cerebellum in a mid-sag section is the white matter is called the arbor vitae, and the gray matter is called the folia, so the leaves on the branches. So that's the basic way you go about doing the mid-sag section. We have mid-sag sections out back there, and they're in a tub that say don't touch. So open it up, look at it. I will also put out, I have a little bit better one. It's a demonstration one, and I'll put that one out as well today. So we get the mid-sag down quickly, and I gave a lot of advice to most of the class individually along the way yesterday. But another good study technique for doing the nervous system is to make a list. What are the structures that you're responsible for that you can see on a mid-sag section? Make that as separate lists, that when you go and study that, you are studying the things you can actually see. Because the other thing I see on lab exams is people, we all make guesses on tests, and but a lot of times I'll see on this test people making a guess, and the guess is something you can't even see in that slice. I mean, it was just not even a possibility on that section. So if you can get yourself so that you, when you look at a section of the brain, that you at least narrow the scope down to the things that you could possibly see, you got a much better chance of getting it right if you guess. All right, so now let's go through the kind of harder stuff. Oh, just one thing. To say something about the annotations. I said this is all annotated, but he uses the kind of true Latin terms, so they're not the Americanized version of them. So you often have to look, you can figure them out. You have to look for the root word. Let's see if I can find one. Like I said, the fornix, right, which is what we call it. There it is. Fornicus, call them fornicus. Well, Septum Pellucidum's good. Medulla's good. What's another one? Maybe I'm thinking of that, but you'll see, like you learn a nucleus called the caudate nucleus, and it's in true Latin it's like nucleolus caudatus something, I can't say. So, but you'll get the point. The annotations are work if you're willing to kind of just look for the root words.
Right.
So let's go back and let's start out with the frontal sections. And what you got in this, one reason I like this website is in the corner here, it's going to show you where the cut was made. Right? And so this is going to move back. And it turns out all the interesting stuff and the stuff you're responsible for is all in this region here. If I cut way out here and over here, I mean, it's important stuff out there, like the stuff in the frontal lobe is kind of important because it's who you are. You lose that stuff and you're a vegetable. But looking at it, all you will see is the white matter and cortex. So we don't have any cuts out here. Because the only thing you can point at is the cortex and the white matter. So all the kind of internal structures are all back here. So they've started their cut here and they stop about here. And we do the same thing. And I want to start by getting a little bit more close to the middle so that I can show you my landmarks and then we'll work the other way around. You want to start here. So, you don't write them down, you're going to hear this shit over and over again for the next 3 days from every time you ask the question. So this is where the rules really start. So first you find what ventricles you can see. What are these? Lateral ventricles. They're the only ventricles that are not on the midline. So they must be lateral. Then I have two ventricles that are on the midline. What are they? 3 and 4. This one here is the third. So these are lateral and third. The fourth ventricle, which is also on the midline, is just in front of the cerebellum. Do I see a cerebellum? So to see a fourth ventricle, you're going to see some part of the cerebellum. So as soon as you don't see a cerebellum in the field of view, the fourth ventricle is not to be seen. So I can see the laterals and the third, and that's the only ones I can see. What does that tell me? That means the structures that I could possibly see in this slice, telencephalon and diencephalon. So you've immediately, by just seeing what ventricles you can see, wiped out the entire brainstem. It's not shown here at all. The next one is a real easy structure to get, the corpus callosum. From here on out, they're a little bit more obscure. You see the folds of the cortex here, and you see this one thing where the cortex folds inward, and this lobe that folds inward's got a name. What's that?
Insula.
Insula. So this is one of my key things I look for. I look for on models, I look for it on slices. So you can see that if I was looking from the outside, I couldn't see that lobe. So it's the one lobe you can't see on an intact whole brain, insula. My other next major landmark is, see this white matter coming down like this? Internal capsule. So there's a few other landmarks I'm going to talk about, but that's my big thing. I want you guys, when you first start looking at these things, find your ventricles, find your corpus callosum, find your insula, and find your internal capsule. Every one you pick up, that should be the first thing you do. Because those are landmarks that are going to be on virtually every one. Not all, but almost every slice. And they're usually very easy to find. And then you find everything else relative to those landmarks. So let's start out and go through some of the other structures. There was I told you in the mid-sag section, there's a membrane that separates the two lateral ventricles. And that's called the Septum Pellucidum. And what did I say was the rule for, what hangs at the bottom of the Septum Pellucidum? I know it was fornix. So there's the fornix. It's hanging at the bottom of the Septum Pellucidum. Works 100% of the time. Next thing to do is I see these, some bits of gray matter that aren't part of the cortex. And what is the general term for gray matter that's not up in the cortex?
Nuclei.
Nuclei. Okay, everybody get that? So in the peripheral nervous system, you have nerves and ganglia. Right? In the central nervous system, you have gray matter and white matter. If that gray matter is out here on the cerebrum all folded up, we call it the cortex. If it's deep in the brain, we call it a nucleus. So we do a handful of these nuclei in here. And I'm going to start with this one right here. And you can see this nucleus is squeezed between the lateral ventricle and the internal capsule. You see that? That's the caudate nucleus. And it is always between the lateral ventricle and the internal capsule. So you show me a lateral ventricle and internal capsule, and there's a caudate. This is very big. There's some other places where it's very tiny. But it actually, she talked about the ram's horn shaped stuff. Well, the caudate follows that, and so does the lateral ventricle. So there's rule number one for finding the caudate, and a rule for finding the caudate that works 100% of the time. Then you'll notice that there's a grouping of nuclei here that's squeezed between the internal capsule and the insula. And it's kind of pie-shaped. And that's a group, that grouping of nuclei is called the lentiform. So there's your rule. The lentiform is always between the insula and the internal capsule, always. Now those nuclei are broken up into groups. We learned one level of grouping there. This one that's closer to the insula, this big guy here, is called the putamen. And these other two nuclei together here are called the globus pallidus. So those are some nuclei of the cerebrum that you need to know. Next thing I go down now, the third ventricle, when we're looking at a mid-sag, there were two major nuclei that, or structures or regions, that I said straddle the third ventricle. Remember the names of those? The thalamus and the hypothalamus. So when I see the third ventricle in any cut, the question I ask myself is, can I see the thalamus or the hypothalamus? And generally I can see one or the other, not both. because in a frontal section, the hypothalamus is more anterior. So it's not a cut and dry rule like the caudate. I can't say I see it in every one, but I can see those things in. So the third ventricle has some gray matter on either side of it. And as you'll see as I go further back, it's very little gray matter, and the third ventricle is pretty tiny at that point. So I know this is the hypothalamus. I'm too far forward. It's small, not much. You're going to see when we get further back, the thalamus is huge, monstrous, and it's more superior. So third ventricle, always think thalamus and hypothalamus and see if it's there. Now here's another structure you will see in most of the sections. It doesn't turn out very well on this picture, though. You see this little, there's something kind of shaped right here. And this is the temporal lobe, here, temporal lobe. And this little structure, if you see it on our slices, when you cut through it, it's all rolled up like a jelly roll. Did you talk about the jelly roll? Mm-hmm. So there's a structure called the hippocampus. And when you cut it in cross-section, you can see that the tissue is kind of folded up like this. And so that becomes a good landmark for, or good character to tell that you're looking at the hippocampus. And I want to make something really clear that people come out of that and they think hippocampus, jelly roll, hippocampus, jelly. That's what it looks like in cross-section. It doesn't look like, we have some cross-sections where it's intact and not cut. It doesn't look anything like a jelly roll. So make sure that jelly roll character you put next to it when cutting cross-section. So that'll be pretty easy to find. And then there's a nucleus that you can see. You see this gray matter right above it? That's a nucleus that sometimes that sits right above this hippocampus in a few slices, because the hippocampus is about that long. So you can make many cuts like this and still see it. The amygdala, which is what this thing is about that big. And it sits out near the end. And so we got like 3 slices over here that are cut in just the right place that you can see the amygdala. So I don't know what it is about the amygdala, unless you're an anger center or something, but I've noticed students want to find the amygdala on every goddamn pro-section in the room. They come up to me, even after I've given this little speech, you are responsible for finding the amygdala on a frontal section, one of these things over here. Nowhere else, don't look for it anywhere else, don't ask me about it. After 7 years of being asked to find the amygdala on everything in the lab, I get a little frustrated. So it's not that I can actually find it on it, but it's really freaking hard unless you have this hippocampus here to kind of key yourself on. But I want to bring that up because now I'm going to start going backwards, and we're going to look and see how the structures change shape, how some of the structures like the amygdala just disappear. Right? But in each case, we want to first find all the landmarks. So this is a little bit further back, and so if you had that thing, what would you do first? Lateral ventricles, third ventricle, right? Insula, internal capsule. Those are the landmarks. You see the caudate's a little bit smaller, but it's still squeezed between those 2 structures. You can still see the entire lentiform. Here's the third ventricle. I'm still just seeing a small amount of gray matter on either side, so I'm still in the hippocampus. I can still see the hippocampus here and the amygdala here and here. So not much has changed other than I can see the pons. So I think by the next slice, things will change a little bit more. And now we'll notice, let me just start out here, well, lateral ventricles in third, right? Internal capsule and insula, my landmarks. I can still see this jelly roll. I can still see my hippocampus, but there's no longer a little thing of gray matter. See, the amygdala's not in this one. I've now cut too far back to see that. Caudate's still in the same place. Notice that I'm seeing less and less of the lentiform. I can still see the putamen over here, but I'm starting to fade on the smaller, more medial structures. I can still see my caudate, I can still see my fornix, but now I've got this large gray mass on either side of the third ventricle, and the third ventricle is a lot bigger. So what is it now? Thalamus. Okay. What else can we see? Well, we can actually see the mammillary bodies. They're part of the very back of the hypothalamus. But this has also got another landmark I like to use. See these white matter coming from the pons going up to the cerebrum. Those are the cerebral peduncles. Another really important landmark when I can see them. The cerebral peduncles are in what vesicle? What embryological vesicle? Or midbrain. I know you guys have been at it for 3 hours. I shouldn't be asking questions. So they're my landmark for midbrain, because everything until I got to this slice was all diencephalon and telencephalon. And now suddenly, here's the structure of the midbrain. Okay, and I'm going to use those in any slice that the midbrain's in. Now there's cerebral peduncles, and that's going to say, oh, if you go look on your list, we do about 7 things in the midbrain. So when you see cerebral peduncles, you should look and say, oh, do I see any of these other structures that are midbrain structures? Other than that, there's not much new on that one. Now you can see, and let's go back, since I trained you guys, lateral ventricles, third, insula, internal capsule, right? But I'm still seeing my cerebral peduncles, and I've cut through the pons. There's a lot more here, but let's go through the ones we saw. There's the caudate, still there. Notice how the lentiform is disappearing. Okay? Still see the putamen on this side, not much else. A little bit, the very end, tail end of the hippocampus there, and now you can really see the thalamus. Here's the third ventricle. Now you cut right through the middle of it. But what's interesting is right in here, this is the midbrain. How do I know? This is cerebral peduncle. And notice that the cerebral peduncle is continuous with the internal capsule. It's going to be a really important thing you need to know for lecture, for lab. Okay? You're going to do pathways next week. One of the pathways you're going to do is something called the pyramidal pathway. This is the pathway from the precentral gyrus, where your motor starts, all the way down into your spinal cord. And the pathway that would have to take, imagine there's a neuron cell body up here, and it needs to have its axon go down into the spinal cord. How the hell does it get there? Well, that axon's got to go right down this path. And what you call the white matter along that path changes. So up in this region up here, it's called the corona radiata. You should put something in your lab manual next to corona radiata. I'm going to give you a special prosection to look for them. Don't look for it there. Then when I hit about here, about the caudate, I now call it the internal capsule. When I get to about right here, which is the end, it now becomes the cerebral peduncle to here. Then it's in the pons, and then it's going to turn into the pyramids of the medulla. So you're going to need to be able to name all that white matter, and you can see it's the same white matter. These tracks here are the same tracks as here, different name. Now, when I look in this region here, in and around the cerebral peduncles, I see some stuff that wasn't there before. So if you go look on your list of other structures in the midbrain you need to know, you'll notice there's something called the substantia nigra, and it's a nucleus in the brain, and it's a little strange because the neurons are pigmented with melanin. It's like the pigment in your skin, so they come out really dark. So these are substantia nigra. Right above it, you see these kind of round nuclei here that are called the red nuclei, red nucleus. So those are things that you are responsible for. And by here, I'm pretty much got the same stuff. Although notice the lentiform's completely gone. Look how small the caudate has become, but it's still there in the same location. Right still in the middle of the thalamus and some of the nuclei here of the midbrain. Okay, so that's the frontal section. So now let's go back and look at a horizontal. I'm almost through, guys. I know let's get rolling. But I'm going to, you can see again here where I've cut. I'm going to bring it down to about here. But the point I want to make is that when I came up with my little set of rules for doing this, my goal was to have the exact same set of rules regardless of whether it's cut like this or this. Because those relationships between those structures don't change when you cut them. So the relationships are the same. So what I'm going to do, even though it's going to look a little different, you always look for the same landmarks. And you don't even, you can get good at doing this and not even have to know which way it's cut. So if you're trying to, a lot of students trying to like, well, is it, I need to be able to tell whether it's horizontally cut or frontal cut. If you can do that, that's good, but you don't have to. So let me zoom down here a bit. I'm going to start with this one here. So the first thing, what do I look for? I look for ventricles. These are not on the midline, so what are they? What are they? Lateral ventricles. Why do I see them twice? Yeah, they go like this. So in a lot of horizontal sections, since the ventricle goes like this and comes back around like that, when you cut into it, that ventricle actually came up like this and came back down here. So a lot of times you see them twice in these ones. And this isn't showing up very well. Right in here, that's the third ventricle, right on the midline. Here's the insula here. Do you see the infolded part here? So there's the insula. And then the last landmark I did was internal capsule. See this white matter here? Now the difference is it always looked like this in that frontal section. When you look down and you cut through them, it's kind of more V-shaped. So again, it definitely looks different, but if you can find those landmarks, and also here's Corpus Callosum coming across here, a little bit of it here. So the Corpus Callosum you often see twice. But those are the same landmarks I found in the other ones, and then I use apply the same rules. Here's the lateral ventricle, here's the internal capsule, what do you call that? Caudate. And that guy follows this all the way up around like this. I'll do it on this side. So it's like this. And you see that little thing right there? What is it? The caudate. So the caudate, it starts out really big and comes, they call this the tail. I've never seen them put it on a test, but I wanted just to point out, you apply the rule, and it works. Right? And then between the insula and the internal capsule, what do I find? Yeah, and maybe not, in this case, not all of the lentiform. Here I can only see the putamen. Relationships are the same. Nothing has changed. Third ventricle, what straddles it? Thalamus. Right? And then this membrane separating these two lateral ventricles is what? So, I hope I made my point there. Is if you write down these rules and you apply these rules, you'll make your life a lot easier. So this is a little bit lower. I'm starting to see more of the lentiform. I can see a portion of the globus pallidus here. Internal capsule, lateral ventricles, third ventricle, insula, corpus callosum. Those are the landmarks you look for. And I'm not going to, we don't do the hippocampus or the amygdala in a horizontal section. So I'm not even going to look for them. In fact, just to get through this really quick, I'm going to zoom all the way down. Oh, that was kind of cool. But let me just, I want to get far enough down where I see this. Look, anybody familiar from your childhood here? Mickey Mouse? Mickey's ears? Okay. So we have a few sections that are cut like this. So what are Mickey Mouse's ears? You may know?
The something
Cerebral
peduncle.
Cerebral peduncles. And the cerebral peduncles are in which vesicle? Midbrain. Okay. So his ears say that this is in the midbrain. So again, then you can go to your list. Once you see Mickey, you can go to your list and say, where does it say midbrain on my list? And then you go, so what can I see? Well, cerebral peduncles, his ears, his eyebrows, substantia nigra. You didn't show up very well, and these are really hard to see, but there's some really faint round guys right here, which are the red nuclei, so his eyes are the red nuclei. His mouth is what? What's that? Yeah, cerebral aqueduct. Yeah. So that's another way. You get away from the Mickey Mouse thing. If I see the cerebral aqueduct, where am I? I'm in the midbrain. So for me, I like to use the ventricles to do that.
What are the eyebrows?
What's that?
What are the eyebrows?
The eyebrows are the substantia nigra.
Oh.
Part of the basal nuclei. And we don't go any lower than this in horizontal sections. We don't. So, and also on the sections that we have that are through the midbrain, ignore the cortex part out here. We're not going to ask you about anything. Here you can see the mammillary bodies, which are actually part of diencephalon, and we do have a few that you can see that. So that's really not a midbrain structure. But when you look at this and you see Mickey, skip all this stuff out here. Okay? What we're interested in is what's going on right in the center of those prosections. Anyway, so that's my speech. I really recommend getting on the slices, do mid-sag, do the slices, and once you got those things down on Monday, we're going to pull out those plastic models, and I'll show you how to use these same rules after you got them down on the slices, how to find stuff on the models. Way easier once you got this.